Okay, so these are the biomedical applications: blood oxygenators, cell washing, plasmapheresis, leukodepressin, dialysis has already been discussed, and then hemofiltration. So that's what we're going to try and get through as close to all of that as possible by 11 o'clock. That's my plan. Okay. We have already talked quite a bit about blood oxygenators. So just to recap, they are actually a little different. So if you think about what we have discussed so far, everything has been pressure-driven membrane processes, right? Ultrafiltration, virus filtration, sterile, all pressure-driven, right? With the one exception, which is membrane adsorbers. And actually, I never discussed this. So what is membrane adsorber? What is the driving force? So actually, it's if you have basically a membrane adsorber with ligands there, right? And there's an attraction that causes them to bind and elute, right? So actually, the driving force is going to be some sort of chemical potential. There's an attraction, there's an enthalpy of adsorption that has also got to be counteracted by the entropy drop, because now it's localized, right? So there's a reaction occurring, but it's a chemical potential ultimately, okay? So we've talked about all those, and then we start talking about blood oxygenators, and the point about a blood oxygenator is it provides a cardiopulmonary bypass during open heart surgery, but it is fundamentally different. It's an example of a gas-liquid contactor. You have liquid on one side of the membrane, gas on the other side, gas fills the membrane pores, right? And you have transfer from the gas to the liquid, and from the liquid to the gas. Carbon dioxide goes the other way, okay? What's the driving force here? Actually, it's a difference in concentration. It's a chemical potential, but it's a little bit different because if you think of it, is this the one? Yes. If you think of it, what you're going to have in a blood oxygenator, if we enlarge this, for example, you have your membrane like this, right? Okay, these are the pores. Okay, those are pores, right? So we have the blood, say, on the outside. So this is blood. We have oxygen, nitrogen on the inside flowing this way. And then here, you are going to have here a gas-liquid interface, right? Because the membrane is hydrophobic, air fills the membrane pores. Now, what we know is that there is an oxygen concentration in the gas phase that does this. You go through the membrane, there's a disconnect, and it goes like this in the blood. And this disconnect is because you have typically a partition coefficient here to explain the fact that here you are measuring concentration in a gas phase, here you're measuring in a liquid phase. Your basis is different, so it's not continuous. Concentration is not continuous, but what is continuous is the chemical potential, okay? So the chemical potential is continuous, and it's going to be lower here, higher here, because you have a higher oxygen concentration here. So oxygen moves this way, and then carbon dioxide will do the opposite, okay? Because actually it's going this way, it's going down. So it's really a chemical potential difference. One could argue also it's a partial pressure difference, okay? Because you can determine the partial pressure oxygen here, determine the equivalent partial pressure that would be in equilibrium with what's in the blood. There's a difference, okay? But more fundamentally, it's a chemical potential difference, okay? But it is different because it involves a gas and a liquid phase. So we talked about that. We talked about the design of these oxygenators. I talked a little bit about the older designs, the more modern designs, about the importance of how you try to optimize them. We rushed through this a little bit, where I basically talked about how we come up with correlations to correlate data. And the reason we do these sorts of correlations is so that we can compare data from many different blood oxygenators, different surface areas, different flow rates, right? Otherwise, you can't compare them. So they are dimensionless correlations that chemical engineers like to use. So we talked about correlations for both flat sheet and hollow fiber blood oxygenators. And then I pointed out that we do also have to be careful, because in the case of blood oxygenators, we also have this sort of oxygen hemoglobin dissociation curve, which we need to include, okay? Because oxygen not only dissolves in the plasma, it also binds to the hemoglobin in the red blood cells. The way we typically, or in these sort of correlations, try to account for that is you come up with a correction to the mass transfer coefficient. All of this stuff here is due to actually the fact that oxygen binds to hemoglobin, so it enhances the rate of mass transfer, right? Because if you didn't have the blood there, the amount of oxygen you could dissolve in the blood, in the water, in the plasma is much less. But since you have blood there, you're actually taking much more because you can absorb some and bind it to hemoglobin. So we are making it, we're trying to model that as an increase in the amount of oxygen you can dissolve in the plasma. That's what we're trying to do. So, and then this E is an enhancement factor, okay? And so I talked about how we could do this. This is the experimental setup. I said typically there's an oxygenation and deoxygenation run in parallel. This is done in any biomedical lab, and you'd use some sort of model blood, sheep blood or bovine blood. And I talked about various solutions that we could use. We talked about membrane surface areas for neonatal and pediatric blood oxygenators. And I think we stopped somewhere about here at the end of these correlations. Okay? All right. So the future of blood oxygenation is actually quite interesting. So I told you that this really, the first procedures are probably in the late 40s, but people didn't have very good survival rates because you had these film blood oxygenators, and sterility was a major issue. So people died because of infection. Okay? Next, we had bubble oxygenators, very popular in the 70s. Sterility is no longer a problem, but now a lot of blood damage. And so it's a trade-off. Do you really want to do this procedure? Because the procedure might be okay, but how will you actually survive because of so much damage to your blood was a question. And then in the 80s, late 80s, we came up with actually early 80s, flat sheet silicon rubber. It's a dense membrane. Now sterility is not a problem, blood damage not a problem. The problem is you don't have good removal of CO2. So gas exchange rates are a problem. Good removal of oxygen, but then good passage of oxygen into the blood, but not good removal of CO2. And so this became a problem of trying to manage the perfusion gases or the gas phase to try and make sure that you get reasonable accounting for this. It turns out actually if you remove CO2 too much, it's also bad. We do need to have some CO2 left over because it helps buffer the pH of the blood, okay? Because as you know, carbon dioxide dissolves in water, forms carbonic acid, okay? And so then the most recent development were these microporous blood oxygenators, which I talked about. And here we have a microporous membrane. There is still a gas-liquid interface, but it's relatively small because it's all these micropores. So you minimize blood damage. You have high rates of gas transfer, okay? And these were used routinely in the late 80s, 90s, 2000s. But the future is moving towards less invasive surgery, okay? So today we would, there are many procedures that in the past used to be fairly, how can I say, very difficult because we have to operate, but today if we can avoid operations, it's good because the patient recovers much more quickly. So imagine open heart surgery. I mean, besides the fact that you might be replacing valves in the heart so that the heart, there's, you know, valves are not working properly and so forth, that's fine. But besides that, you're cutting open the patient for however many, 2, 3, 4, 5 hours, has to be living on blood that's being pumped around an outside circuit. There are high amounts of transfusion required. Transfusions are always risky because there is the risk of infection from the transfused blood. Okay, blood is safe, but it's today, but it's still a risk. And the other thing is there has been shown to be many side effects due to cardiopulmonary bypass, which are more psychological. So people tend to get very depressed. They have various. Then this passes, but there are all these side effects. Okay. And so the idea is, can we do it by, you know, just putting very small incisions and then using a laser and using these sort of robotic methods? And that's done quite a bit today. And also what is more, if you can do it without stopping the heart from beating, that's even better, okay? The whole problem is here you had to stop the heart from beating because a surgeon can't go and start operating if the heart is moving like this all the time. But today there are beating heart procedures, which is much better because then the natural lungs can keep working, everything is working. So because of that, the demand for blood oxygenation has dropped, okay? You still need it for certain cases, but overall the demand has dropped. However, there are a few other things happening. So there's extracorporeal life support, ECLS, for extremely preterm infants, say about 28 weeks. So in that area, in the past, you know, many of these, if you're for that early, many of these babies didn't survive, okay? But today there are possibilities to try and keep them alive, but you have to be able to help them get oxygen because the lungs are not developed enough, okay? And so again, there's a great deal of interest in using this sort of blood oxygenation to provide oxygen. So this is one area of development, okay? The other is something called an intravascular oxygenator or an IVOX. So in cases where patients have surgery on their lungs, so I should point out that doing any sort of surgical procedure on the lungs is tricky. The rates of survival are not high and it's not that easy, okay? It's not easy, but there are cases when that's done, and it takes a little while for the lungs to heal and come back to a sufficient capacity. You'll never get your full capacity, but capacity for you to walk and so forth. In those cases, you can provide sort of mid-term support. So basically, say for 3 months or so, you try and provide these patients with an additional source of oxygen. So the idea is quite simple, actually. Imagine you take these hollow fibers, okay? So you take hollow fibers like this. You seal them at one end, so they're not flow through. So you've got a whole lot like this. They're microporous, okay? And they are attached here, and this goes. So this is inside a blood vessel, okay? So this is some sort of blood vessel. You have attached them here, and then this goes outside the body. So it's got to be sterile, though. That's the problem. But you pass oxygen, nitrogen in here, and it can diffuse directly into the blood, right? And carbon dioxide can come the other way. These are called intravascular oxygenators. They are used, they are designed, they exist. They're tricky because you've got to make sure you keep sterility, okay? You don't want any sort of pathogens getting in, but they work quite well, okay? They're different to the blood oxygenators we just talked about because this time you want these to last for about 6 months, okay? Up to 6 months. The blood oxygenator was only 6 weeks. I'm sorry, 6 hours. 6 hours, right? 6 hours. These can be much longer. So much more concern. See, in a blood oxygenator, I told you, you have a hydrophobic membrane, but eventually proteins foul, adsorb, and you start to get leaking. So this is not allowed to leak. If it leaks, there's a problem, okay? So typically there's a lot of surface coating, surface modification of the membrane, okay? And then the last one is the surface modification to prevent emboli. So that is something that remains a problem in blood oxygenation. And let me see if I can go back to my very first or second slide. Yeah. So you know when you're, when you're going through this procedure, you can imagine you're taking blood out of the patient, okay? It's going through this venous reservoir, through a blood pump, through the oxygenator. There are various filters all over the place, by the way, filters to remove emboli or blood that's clotting, okay? It comes into the cardiotomy reservoir. From there, it comes into the self-salvage if we're having it, collection bag, reinfusion bag, and typically a lot of anticoagulants are added because you don't want the blood to start clotting. And remember, blood clots, right? When it's a couple of things. When it's exposed to air, it starts to clot. That's a natural reaction. When you have high shear stresses on these, you can start off the clotting cascade. You want to stop that, so typically people are given heparin and other sort of anticoagulants. But what can happen is you can still get these microemboli, which is a few cells that have gathered together and aggregated and form an embolism, okay? These are very dangerous because they can then start going through the body. So there are filters all over the place to try and remove them, but nevertheless they can get into the body. And if they do, I mean, they can pass through the aorta and big blood vessels, that's fine. The risk is that they go into the brain. That's the biggest risk, because if it gets stuck in one of the blood vessels in your brain, you can get a tumor. So that's very serious. They can also go into your legs and other places, and again, form clots that can block things. So this is very important. So there is a great deal of interest in coming up with methods to try and guarantee against not getting any sort of embolism. That's a major concern in blood oxygenation. And one way is people use actually silica. Silica actually has properties that help break these up, and that's one thing. But the problem is you don't want silica, even nanoparticles of silica going through your body. And so there's a lot of research in this area to see if we can come up with better surface coatings on these oxygenators and all the tubing and everything else that would prevent the occurrence of emboli. Okay, so that's another area of development still in this topic. Okay, so I think that's all I want to say about blood oxygenation. If you have questions, you can always email me, but it is a very mature technology. Okay, let me talk now a little bit about washing red blood cells, or the general idea actually of cryopreservation of cells. So today there's a great, actually it's routine to preserve all sorts of cells, right? The cord blood, when you're born, the blood in the umbilical cord and in the placenta, if you can recover that and collect it and keep it, you've got a lot of hematopoietic stem cells there. These are the progenitor cells that will lead to all sorts of cells, blood cells in the body. And so they're very valuable because they are your own, okay? So if you can keep them and freeze them, then if you ever have cancer or something like this, you can always, kill off all your bone marrow, I'm making it extreme, and reintroduce these cells, and they should be able to repopulate your immune system, your blood cells, everything. So, I mean, people do freeze cord blood. Today there are blood banks all over the world that are doing this, okay? That's one example. Another example here that I was going to talk about is washing red blood cells. In the US, this is used a lot by the military. I think it's used in Europe also a little bit. The idea is, but for civilian applications. But the idea is this. We donate blood, right? And when you donate blood, you can donate whole blood. We'll talk more about this in plasmapheresis. But you can also donate fractions, or you can take the whole blood and fractionate it. And so what are the fractions? You end up with plasma and plasma proteins. You end up with red blood cells. You can end up with platelets, okay? And typically leukocytes, white blood cells are useless, okay? So you throw them away because my white blood cells are going to interact with yours, with the next person, They're very, very specific. So you can't give white blood cells from one person to another. Red blood cells you can match, okay? You can match them. Now, in the case of red blood cells, if we just focus on that, the red blood cells have a lifetime of about 120 days. So after 120 days, they die. And they're in your body. They don't have a nucleus. So they flow around, and eventually they're removed by the spleen. And then the hemoglobin is recovered, and you're making new cells, and so forth. When you take them out of the body, there's a certain lifetime, okay? But after 3 months, they're all dead, okay? Just assume they're all lysed. So you can keep them maybe 45 days or something like this, after which their therapeutic value is very low because too many of the cells have lysed. And so firstly, there's a problem. You've got free hemoglobin. Turns out that hemoglobin is actually very dangerous. It's very reactive. That's why if you take your blood, actually in your blood, there is no free hemoglobin, okay? The body has developed mechanisms that if a red cell lyses somewhere in the body, the hemoglobin is immediately reacted and taken away, okay? Because hemoglobin is highly reactive. But the red cells have hemoglobin inside them, okay? So you don't want to infuse people with lots of free hemoglobin. That's a very bad thing. So as you wait, this blood is lysing. That's not a good thing. Secondly, there are not that many viable red cells. So again, it's useless, okay? There's only a certain lifetime, and you store them at 4 degrees Celsius. So this initially was for military operations. If you suddenly have a war, you need red blood cells very quickly. So how can you get them? So one way is if we got all these donations early on and froze them, then when you need them, all you've got to do is thaw them, and you've got your red blood cells. And the shelf life now is about 10 years, okay? So this is a way of storing them. In Europe, there was a concern, and the concern was this, especially in the 80s, 90s, when we're very concerned about, AIDS and other things that you can get from donations, is that I can donate my own red blood cells to myself, right? So I donate them, freeze them, keep them. If I need them at any point, thaw them and use them, okay? So there are civilian applications also. So that's what I was going to talk about. That's an example of cell washing, but there's that. There is, I talked about hematopoietic stem cells. So there are many sorts of cells here that people try to preserve so that you can have your own cells, okay? Now, the thing about it is you can't just take cells and freeze them, and you freeze at about minus 80 degrees Celsius, because what happens, water freezes, as it freezes, it expands, it will burst the cells, and it's finished. So you need to put a cryoprotectant, something that basically, if you like, exchanges for water, does not expand, allows you to freeze the whole thing, so that then when you thaw it, the cell is still viable. That's the point, okay? And that is why membranes come up, because if you add the cryoprotectant, you have to take it out. So we've talked about this before. This is how you would operate tangential flow microfiltration, and this is a microfiltration process, okay? So you could either do a concentration mode, or this is actually total recycle. The feed's coming, you take your retentate back and your permeate, you put it back in the feed reservoir. So typically we do that for lab scale experiments to look at membrane performance under constant concentration. Batch concentration is very common in the biotech industry and also in this cell washing. Sometimes when you want to concentrate blood, you remove the permeate, okay? We've talked a lot about diafiltration, so you're familiar with the diafiltration process. And then this final one called feed and bleed, where basically if you look at the feed, some of the retentate comes back, but some of it is removed, okay? So these are modes of operating, and it's all in this book. which I will give you the reference for, okay? All right, so I'm going to talk about removal of cryoprotected from frozen red blood cells, and this is some work we did a long, long time ago. So basically, there are three sorts of cells we look at: red blood cells, stem cells, and platelets. Okay, so platelets are used for clotting. Okay, and the cryoprotectant is glycerol or dimethyl sulfoxide, DMSO. Those are very commonly used cryoprotectants. Okay. So the shelf life for red blood cells, as I said, is up to 10 years. They can be deployed in various depots and no complicated logistics in recovering them. Now, one thing about it is that the post-thaw shelf life, after you thaw them, was limited to 24 hours. The equipment was heavy, they were floor mounted. So the idea was, could you come up with a membrane system that's very lightweight? And if it's closed and sterile, you should then be able to keep them for however long the cells are viable. Now, cells that have been frozen and thawed are not as healthy, if you like, as cells that you just take out of your body now. So the shelf life will be less than 45 days, but maybe 3 weeks, which is much better than 24 hours. Okay, and so these are the processes involved. You have to first you're frozen them. They're at minus 80 degrees Celsius. You have to thaw them. You have to pre-dilute, and then you have to wash. So why do you pre-dilute? Well, I think everyone's familiar with osmosis, right? So if you've taken these red blood cells, you've frozen them in, say, 1 molar glycerol, something like this. After you thaw them, you now want to slowly take out the glycerol, okay? But you need to pre-dilute it first. Otherwise, if you suddenly start bringing it in touch with what's called isotonic saline, so that is saline at the same osmolality as our plasma, okay, you will burst the cells, because they'll have a very high glycerol concentration, a much higher osmolality inside than outside. Everything will rush in, the cells will burst. So that's why you pre-dilute slowly. And then you wash to remove glycerol. There is also some hemolysis that occurs. You must remove the hemoglobin, and you want to concentrate them to a hematocrit that's compatible with the body, so probably about 25–30%, okay, by volume. Okay, so this is just to give you some idea of the sort of volumes. The volume of a unit is about 800 ml. This is diluted. Okay, when you donate 500 ml, we heard, but this is diluted. And there is significant variation actually. Hematocrit is low, 15%. Glycerol concentration after thawing is 1.57 molar. This is a very high supernatant hemoglobin concentration. Okay, 540 milligrams per deciliter is very high. That means there is hemolysis. You've got to remove that. You cannot inject that into the patient. And this is what we want to do after washing. We want a hematocrit of 40%. We want a glycerol concentration of 0.1 or less. And the supernatant hemoglobin should be less than 200. Okay, that's what we want. So we basically come up. So if you look here, we have to concentrate because we are starting off at a hematocrit of 15%. We've got to get to 40%. We've got to concentrate, but we also have to do a diafiltration. We've got to do both steps. Okay. And really, what this amounts to then is these really series of operating. Oh, I want to show you this. So this was something that we talked about during microfiltration. I mentioned this idea of laminar flow for those of you that don't know about laminar flow. So laminar flow basically is the following. When you flow, and typically we talk about a Reynolds number of less than 2100, you have what's called this parabolic velocity profile. Okay? So fluid here is moving faster than closer to the wall because the wall provides some sort of drag force or restraint. Okay? And so that's what's meant by laminar flow, and with it, we still have flux, which is the volumetric flow rate divided by membrane surface area. Transluminal pressure drop is P0 minus PL. Wall shear rate is what's happening at the wall, the shear rate at the wall. Transmembrane pressure, these are definitions we've talked about. Okay? Okay, so let's move through this. So these are some mass balancers. So let me just explain briefly how I got to this. But if you're not sure, that's also fine. What happened here? Come on. Okay, so what you can do is you have your blood bag here, okay, and you can do what we call a mass balance around the blood bag. My writing is not clear. Okay, never mind. So we basically say accumulation equals in minus out. That's typically how we do a mass balance, okay? So in the blood bag, the accumulation is given by this term, dCT dt multiplied by Vb. The volume in the blood bag multiplied by the change in concentration with time. That is the accumulation term. Now, there's no glycerol coming in because the diluent is isotonic saline. But there's glycerol flowing out, and it's going out at the concentration in this bag. We assume the bag is fully mixed, multiplied by the permeate flow rate. Okay? So you can do that, you can integrate it, and you find that the variation in glycerol concentration is an exponential decrease. If you remember when we talked about diafiltration a couple of days ago, I told you the concentration goes down exponentially, right? This is just proof of it, and if you're inclined, you can do it, calculate it yourself, and if not, it's just the way it is. You can also write in terms of dia volumes. This Vt actually on Vb, that gives you the dia volume, right? Number of dia volumes. Okay, so that's fine. We can do the same thing for hemoglobin, okay? And so this I did many years ago when I was actually working in industry. Why didn't that move? It did. So I thought it would be interesting to give you an actual real problem. So we made hollow fibers with 163 polysulfone fibers, 430 centimeters squared membrane surface area, 14 centimeters in length. The inside diameter was 600 micron, outside diameter 1000, 0.22 micron pores. So they are fairly large fibers, okay, because you want the red blood cells to flow there without getting plugged and so forth. Wall sheet of 2000 reciprocal seconds, quite normal, and the transmembrane pressure drop is not very high, 80 millimeters of mercury. Now, here are the conditions. We wanted to make sure that the whole diafiltration step could be done in 30 minutes. So we've done a problem like this, right? So you can size your membrane surface area and so forth. But there was another thing. We didn't want to use more than 1.5 liters of diluent. So think about this. The amount of diluent you need, it depends on how many dia volumes we're going to use. The volume of one dia volume depends upon the blood volume in the feed. So we are starting off with around 800 ml at a low hematocrit, but we know we've got to end up at a hematocrit of 40%. So there's an interesting optimizing problem here. Do you do the following, right? Do you, so do you start off here and do diafiltration first, okay, at 800 ml blood volume, and then concentrate to whatever it is, and actually it's about, I think it's about 540 ml, I forgot now, 540 ml, something like this, okay? So do you do the diafiltration first and then concentrate? So what's the advantage of this? Well, if you remember our flux equations, the lower the cell concentration, the higher the flux. Processing time will be shorter, okay? What's the disadvantage? If we are using, say, 3 dia volumes, you need 3 by 800 ml of saline diluent. If you do it here, if you concentrate first, what happens? Well, the amount of diluent you need is less because your feed volume is less. Your flux is lower, okay, because the cell concentration is higher. But on the other hand, the amount of liquid that you have to process is less. So even though the flux is less, the time may be less, right? You don't know. So there's an interesting or maybe there's something in the middle where you concentrate a bit, then you conduct diafiltration, then you concentrate some more. But there's an optimization problem here, okay? And actually this applies to the biotech industry as well, okay? So this was quite interesting, I thought. Okay, so we move on. And so this is just some experimental data. This is our variation of glycerol concentration with dia volume. So 1, 2, 3, 4, 5. And what I'm trying to say is we got very good agreement, okay? So that's actually pretty good agreement. When you do it for hemoglobin, you don't get such good agreement. You find, in fact, the experimental data lie here above what we predict. So what does that mean? That means there is some rejection of hemoglobin. Now that would be strange because these were rather large, 0.22 micron pore size membranes. Hemoglobin is tiny. So what it means is that there are some lysed cells that are going and getting stuck on the membrane, and they are rejecting hemoglobin, okay? So it means that hemoglobin, therefore, is the removal is going to be a little less than predicted if there were no rejection, okay? All right, so this is what I told you. You want to minimize processing time. You could conduct diafiltration first, high flux, high diluent volume, or conduct diafiltration last, lower flux, lower diluent volume, which is optimal. So actually, if you're interested in the mathematics of this, you can send me an email. You can actually solve this analytically and show that the optimum hematocrit at which to conduct, or cell concentration at which to conduct this, is going to be somewhere around here. These are the calculated or the model curves, okay? And you can understand why there is this optimum, right? Because on the one hand, you have a very large diluent, high flux, but it takes a long time to process all that diluent. On the other hand, only a small amount of diluent, low flux, but the time is less again, okay? But at some point, there's an optimum, okay? And in fact, this optimum turns out to be, you can show that it's the wall concentration divided by E. But if you're interested in doing the analytical solution, let me know, and I can email you the way to do it, or email you a paper. There are a couple of papers, and our paper also calculates it, okay? All right, so anyway, we then went and designed the system to finish the story. So this was our prototype, and this is typical of a scale-up, but this was a fairly easy scale-up, because this is what we did in the lab, our prototype, okay? And then we wanted to scale up because we want to get our time down, because this wasn't doing it in 30 minutes. So we had to scale up, and we basically changed our area from, I'm sorry, this was the prototype, this is the scale-up one. We changed the area from 430 to 1720 square centimeters, okay? So the scale-up parameter was to change the area. We kept the wall shear rate the same because we don't want more blood damage. The feed flow rate we changed marginally, that was okay. And the diafiltration time went from 90 minutes to 20 minutes. And the time for concentration step was 10 minutes approximately, okay? And we also changed the length. You can see here, the prototype was 14 centimeters. We used much shorter modules. Why? Remember I told you, the longer the module, you get more concentration polarization, average flux decreases, okay? So that's what we did, and I think I can end this part by showing you the actual unit. This was many years ago, but I thought it was a nice, yeah, so that's the unit. And you can see here, this is old technology. Today you do it much better. But anyway, for the time, this was automated. Here is the membrane, the hollow fiber module. So today you could do this in half the size and you could automate it. But anyway, that's a hollow fiber module. This was a pre-filter. You needed to remove any aggregates. Here is our waste bag. So the waste, glycerol containing waste goes in here. Okay, this is our diluent, all right. We also add something called adjuvants. At the very end of the process, you add these adjuvants, which basically helps preserve the red blood cells, okay. So it's like adjusting some of the salt species and so forth present. The other interesting I want to point out, again, this is all old technology, so we have a much better valving system today. But if you look at all this is one big tube set. It's all tubes, all sealed, all sterilized. You just hang it on there and you run it. And that's what people like to do in the biotech industry today with single-use devices. This is a single-use hollow fiber module, okay? So I think that brings me to the end of the cell. I might have one or two more slides. Let me see. That may be it. Let me see. Okay, so we did some work also with stem cells and platelets. You can see the volumes are quite different, and this time we're removing DMSO. But again, we get excellent agreement, okay, with our membrane. So this is removing cryoprotectant. So microfiltration, ultrafiltration can be used effectively for buffer exchange. Many applications in biomedical separations. Buffer exchange occurs frequently in downstream biopharmaceutical products. So I think that brings me to the end of that lecture. So I just want to summarize that this idea of cell washing, I thought it was nice to show you something that used microfiltration. You could also use ultrafiltration. And there are many other examples as well, besides stem cells and red blood cells, okay? So I think we're doing okay. It's 10:30. So let me now, as I said, move on to the second lecture, which is the other part of this biomedical processing. And I think that at that point we may stop if it gets to 11, and then we can do the rest after that. So let me get to the next lecture, just 1 second. Any questions on this one, by the way, while I get the next one? Yes. So any questions on that part? I think the same principles of microfiltration apply, slightly different concerns because we're working with human blood. By the way, this is also now, people are now looking at these sort of things for veterinary applications as well, okay? So, okay, let's move to the second part then. And again, it seems in this course, partly it was a matter of time, but partly the majority of it dealt with bioseparations. But as you can see, the biomedical applications we talk about here really are very much based on the same theory, okay? So it's a good fit. So we've talked about microfiltration. Blood oxygenators were gas liquid contactors. We talked about them. We talked about microfiltration applications, washing red blood cells. But another one is something called plasmapheresis, okay? So let's talk about that next. So has everyone heard of plasmapheresis? That's the first point. Everyone heard of plasmapheresis? Know what it is or not? No. Okay, so let's discuss that. So you know, and I don't know what's done in India, but in the US, typically people go and donate red blood cells, okay? And they do that by donating what you call whole blood. So you go, you sit there, you lie down, they take out 500 ml of blood, and you go away, okay? And you can do that every so often. That whole blood in the US is then typically taken and people decide what to do with it. It is rare for me to get a donation or a transfusion of whole blood. That is very rare today. It happens, but it's rare. Because typically what you want to do is give the patient the blood component they need, not just everything. And that has a lot of good reasons for that, because you reduce the risk of infection, you reduce the risk of any sort of reactions and anything else. So the main fractions from whole blood would be plasma, so blood plasma. People need plasma sometimes. You could also try and recover specific plasma proteins, clotting factors, and so forth. So that's one area of processing. The other is red blood cells. So, you know, if you're shot, if you have surgery, anything like that, if you're bleeding and you suddenly need blood, you need red blood cells because they are the ones that will give you oxygen, okay? So we do something called red blood cell concentrates. So we take the whole blood and we spin it typically in a centrifuge, okay? And when you spin it, you start off with a red blood cell concentration, depending on the patient, maybe 30%, 35%, you increase it to 80%. Okay, you remove all the plasma proteins, you resuspend the red blood cells in isotonic saline, and that's your red blood cell concentrate. So then if you need a transfusion, you get red blood cells. Typically, you've taken out the leukocytes because the white blood cells actually, as I mentioned, everybody will have an immune response to another person's white blood. You cannot match white blood cells. Now, typically, we don't worry about it that much because if you're healthy, the number of white blood cells you're getting is small. But remember, you wouldn't get a transfusion if you're healthy. So that is the catch, right? So you're unhealthy, and so this could be a big problem, okay? And then the other thing is platelets. So platelets are tricky. There's been a lot of work done on trying to recover, freeze, and reinfuse platelets for people with hemophilia and things like this that, you know, are bleeding all the time. It is done, but it's very tricky because platelets start to get activated very easily. But that's another blood component, okay? So that's one way when you donate whole blood. The other thing you can do is you can go and just donate your plasma only, okay? And this is much more common in Europe than in the US. Because in the US, if you donate plasma, you are paid for it. If you donate whole blood, it's free. It's just a community service. You donate your blood. So you could donate plasma and then you're paid for it. Now, the thing about plasma is if you donate plasma, you can donate more often, okay? If you donate whole blood, your body takes time to make more red blood cells. So you've got to wait a bit. But with the plasma, it's quicker to donate more often. So if you want to, you know, make money on this, because, you know, it's usually poor people and so forth who are donating plasma, you can donate more often, okay? So these are two different things. So plasmapheresis then is part of these apheresis techniques where you're trying to recover plasma and do things with plasma, okay? Now, apheresis, a specific component of the blood, plasma component, white cells, red cells, platelets, is removed and returned, and the rest is returned to the patient. So that's what apheresis, take a specific component, right? If you're talking about plasmapheresis, you're just taking plasma. You're returning everything else, as opposed to cytopheresis, anything else, okay? Now, there are two types of plasmapheresis. There's therapeutic and donor, actually apheresis. So donor is when I'm quite healthy, but I'm donating a certain portion of my blood, a certain component. Therapeutic is if I have a medical condition where this has to be done as part of a treatment, okay? So there are two applications here. So plasmapheresis, therapeutic and donor, refers to removal of plasma. Cataphoresis, selective removal of one or more cellular components. It's usually therapeutic, okay? You donate red blood cells, whole blood, you donate plasma. You don't donate just red blood cells. That's impossible, okay? Now, it's required to separate blood component can be accomplished by centrifugation or membrane filtration. Here the focus on membrane processes which may or may not be the most widely used but have potential advantages. So I want to make this clear. Because this course is about membrane processing, I'm talking about membrane processing, okay? But plasmapheresis in the past, and I think even today, the majority of it is done by centrifugation. So you centrifuge, the red blood cells are more dense, they come to the bottom, you express the plasma, and so forth. But there are many advantages to using a microfiltration process where you can concentrate red cells and return plasma. And it is done. It's not that it's not done. It is done. But routinely, it tends to be centrifuges, okay? But there are advantages. Okay, so use of microporous membranes to separate red blood cells from plasma actually was proposed in the 1940s, a long time ago. And that goes back to about the time when we started to have commercial microfiltration membranes for the first time. We didn't have a biotech industry, so there are no biotech applications, okay? This we learned about a few days ago. Development of tangential flow filtration methods of operation occurred in the 70s. It was an offshoot from reverse osmosis, which also occurred in the 70s, and provided a way to avoid fouling and plugging that occurs in normal flow filtration. So initially, people were talking in the 40s about using plasmapheresis. It wasn't called plasmapheresis, but to remove plasma using depth filters. Doesn't work very well because all the red blood cells get clogged up and caught up in the depth filter. You will never recover the dead red blood cells. So basically, you're wasting them, okay? Tangential flow filtration, you will recover them and you can reuse them. Membrane pore sizes that people use are typically 0.2 to 0.6 micron. You need to ensure high flux because you want to minimize the processing time. Whether it's therapeutic or donor, you don't want the patient to be there forever, okay? It needs to be quick, okay? So 30 minutes, 40 minutes, whatever. You also want to minimize volumes, right? You don't want a lot of wasted volume because you're using plasma then. And minimize hemolysis of red blood cells because that's pointless. If you're hemolyzing red blood cells, you can't give them back to the patient. Theory of microfiltration, sorry. As discussed in earlier lectures, back migration here is dominated by shear-induced diffusion, okay? So in this, remember we talked about Brownian diffusion, inertial lift, shear-induced diffusion? This is a case where shear-induced diffusion is the major back migration mechanism, not the others. So you can model this very well with shear-induced diffusion, okay? Okay, another spelling error here, sorry. A number of methods have been developed to selectively remove specific toxin or immunogenic compounds from the plasma. Treated plasma is returned to the patient with cellular components. So that's one application, okay, where this process called plasma perfusion, where the idea is now it's more specific than just trying to take red blood cells and plasma. It's just taking the plasma itself and trying to recover specific or remove specific components from it, okay, and return the rest to the patient. Because again, the more of your own blood that you can return in therapeutic applications, the better, right? The more you lose and waste, it's not good because your body has to produce it again. So that was the aim there. So plasma perfusion, online plasma treatment performed using membranes or sorbent-based systems. Membranes operate by size exclusion, remove proteins based on size. Selection of membrane operating conditions is critical to maximize passage of specific species to be used, yet reject undesired proteins. And you can also use something from cryofiltration and thermofiltration. So let's just summarize this. So if we now look at just the plasma component and it's therapeutic, we want to remove a specific component in the plasma. So you've got, and there are many, many conditions here, okay, where you have things in your plasma that are causing various diseases, whatever. You want to remove just this one component. So you could use an adsorptive resin where you'd bind this component. But you could also use a membrane if there's a big size difference. If this component is rather large, then you could let everything else go through and reject it, you see. And that happens sometimes. And so a membrane process would be better than an adsorptive process because one thing about packed beds and packed columns is if you adsorb, there's a possibility of interaction with other species. And that can always lead to damage, changing of surface properties. You're talking about very fragile proteins and things, okay? So if we can use a size exclusion, it would be better. So membranes are considered for plasma perfusion, okay? That's one. You have to be very careful about operating conditions because any sort of membrane fouling means you could start to lose components that you want to go through, right? So your recovery is low, and that would then not be ideal. And the final thing is, when these processes are used, what temperature should you use? So we discussed this yesterday under blood oxygenation, and we said that the problem is your body is at, 38 degrees Celsius, something like this. If you're running at room temperature, you're putting a big strain on the body. You're going to get hypothermia at some point, okay? And so there has to be some temperature control, but maybe you can play with this. Some of these components that you're trying to remove may not be very soluble. So if you drop the temperature, they may actually precipitate. And if they do, then they could be very easy to remove, okay? And then if you've got a precipitate, you could easily use a microfiltration, ultrafiltration membrane to remove a precipitate, Okay, so that's what's meant by thermofiltration, basically by cryofiltration, sorry. So you drop the temperature. Thermofiltration, opposite, you increase the temperature. Either way, you can do this. Just remember though, you have very fragile proteins there. You cannot increase the temperature much more than about 39 degrees Celsius. You'll start to denature them, okay, and that's not good because, we can't have very high fever or we die at some point. Likewise, you can't go too low because you might actually start to aggregate these ones that you want, and then when you go up in temperature, they may actually come out in a different conformation, and that would be problematic, okay? But this is done clinically, okay? All right, so here are some diagrams to summarize this part of it. And so here you can see, here's a plasmapheresis or a plasma filter, plasma separator, and you can see that we can remove components that we want, and we can pump back the rest of the patient. And typically the membrane looks like a microfiltration membrane where you can remove components you don't want. Now, one thing to bear in mind, typically in these processes, you remove a component you don't want, you often have to add something back in because you don't want to change blood volume. So you would add things like, say, amino acids, saline, depends on the patient, okay? But something that's beneficial, okay? Apheresis techniques are unique in the range of metabolic disorders that they are treated, okay? It's really quite amazing because if you think of it, blood is like the highway. It's a circulatory system in the body, takes things to cells, takes them away from cells. So there are a huge number of diseases that basically, if you treat blood, you can treat the disease because the blood is the carrier of these things, okay? Donor plasmapheresis is also an important application. Centrifugation frequently used, though membrane processes also exist. So you've got to think of this is actually a very a mature technology, this area. Donor plasmapheresis is routinely done. So what would be the areas of development here? And these are as follows. People are concerned about the cost of healthcare. That's increasing. So cost here is a very important point. If you can come up with a cheaper cost system now, that would be great. People would buy it immediately. Blood banks would buy it immediately, okay? That's one thing. The second thing, though, is a plasmapheresis system, if you can simplify it, it would be good. Because actually, in blood banks, anyway, in the US for sure, typically you have technicians working there who may not have the highest level of education. So you don't want them to basically have to do complex operations. Ideally, you take this unit, you hang it up, you start it, it runs, it stops, you take it out, you throw it away, you put the next one. You have a disposable system, ideally, okay? So that's another thing. If it can fit in the practice, it's good. Now, the last thing is, especially Africa, other places, therapeutic plasmapheresis is complicated because you need a power supply. You need to run all this, right? And if you don't have a reliable power supply, there's a problem. And if you're in the middle of the desert or something else, how would you do this? If you want to do blood donations in a place that's very remote, how would you do it if there's no power supply? So the idea of using membranes and gravity flow is something that's quite interesting. So the idea is as follows, and this is something that there is quite a lot of work going into it, and there are systems that have been designed as well. So imagine you have a patient here, this is a patient, lying on a bed, so I don't know, maybe that's 1 meter in height, and here's the patient, okay, lying there. And the patient has basically, if you like, there's a pole here and a blood bag here. So initially you run your plasmapheresis where the blood bag is on the ground. Maybe this is 1.5 meters. And this is the head you have, okay, for removing plasma, okay. So the system runs like this. This is the head you have for removing plasma. You finish that and you put it up here and then you run it again this way, right? You put the plasma and the red cells back in, okay? And in each pass, you can remove some plasma. So basically you're using, it's a microfiltration step, but your pressure driving force is the gravity head that you have. And you can do it twice, right? You can do it once where the blood flows in and once where it flows back out. In each case, you have this attached to a permeate. So you're taking red cells out and then you're putting them back in again, okay? So that's one idea of what's called donor plasmapheresis using that avoids a centrifuge, right? A centrifuge is what's routinely used, but centrifugation does require a power source. So that's one idea, okay? So like that, that's one thing. You could also use membranes, you know, in a city area with a power supply and so forth. The problem there is that membranes might be more gentle, but since centrifugation is well established and everyone's using it, unless there's a big driving force for changing, nobody wants to do it, okay? But those are ideas, okay? Okay, so the last one I want to talk about here in a way of microfiltration is something called leukodepletion. So, I don't know if you realized it, but if you go and have to get a unit of red blood cells, so I don't know what happens in India, but in US, there's at least 90% of them are leukodepleted, okay? So what happens is you go, this is now for donor red blood cells. You go, you donate your red blood cells, you have a bag, maybe 500 ml, and then the first thing they do is once they collect this blood, they cool it, they send it off to the blood bank, and in the blood bank, they leukodeplete, okay? So they run it through a filter. It's a depth filter, and the idea is to remove leukocytes. And there are various regulations. In the US, you have to remove at least 3 log or 1,000-fold depletion in leukocytes. And then you can do other things. You can end up with red blood cell concentrates, you can remove the plasma, whatever, but you take the leukocytes out and you throw them away, okay? Because as I said, you can't transfuse leukocytes. And the presence of leukocytes means that the amount of side effects or sort of immune response you get will be greater, and there is supposedly a risk, since leukocytes are part of the body's system, right, to prevent disease. They go and catch viruses and things. There is a risk that you may be infecting the patient with viruses if they are still viable, okay? So leukocytes can distinguish between body's own cells and foreign or allergenic cells. During allergenic blood, that means if I give you a blood transfusion, not my own blood, during allergenic blood transfusions, the patient receives allergenic donor leukocytes that are recognized as foreign cells, which lead to several adverse reactions. This may be avoided by leukodepleting donated whole blood and red blood cell concentrates. And basically, we've learned about this. What you use is a multimodal filter, okay, a multimodal depth filter. So actually, it's made typically of cellulose fibers, just like the depth filters we've seen for biotech applications, but with different design criteria. So you want red blood cells to pass through. You want leukocytes to be removed, okay? And you don't want plasma proteins and things adsorbing on the surface. So they should be non-adsorbing. Remember, red blood cells are very flexible. White blood cells are not so flexible. So you'd like to have a porous structure where the red blood cells can, you know, deform and go through, but the leukocytes are trapped. That's size exclusion. In addition to that, you could put specific markers and agents that adsorb white blood cells and allow red blood cells to go through again. Again, red blood cells have less surface markers on them, so they could just go through. So there are many, many different ways this could be done. This was just to show you, oops, I need to, sorry. Okay, so this didn't come out so clearly, but this is to show you there are many sorts of leukocytes. Okay, there are lymphocytes, leukocytes, granulocytes, and so forth. So there are many different sizes. These can go from say 2 micron to about 20 micron. So just size-based won't work. Okay, you need multimodal, and this is what a typical leukocyte filter looks like. Okay, you attach it at one end, blood flows through here, and your red blood cells come out the other end. Okay, that's typically how it runs. But as I said, they are multimodal filters. That's all they are. Let me go to the next one. Oops, sorry. Okay, and so this is typically how they run in a blood bank. And again, the motivation or the design criteria here are how quickly you can do this and how cheaply you can do it. So you can imagine all this donated blood comes in, okay, and there's a technician there, and he or she hangs them all up in a row, attaches them, and lets them run. So if you start running this one first, then you go to the other end of the whole thing. So you start by hanging up this one, and ideally go all the way to this, and then by the time that you come back here, this is done now, okay, and you take it out and put it away. Here are some of the mechanisms. So you can have blocking, where basically the leukocytes can go and get blocked, okay. You can have bridging, where 2 or 3 of them attach together. They do aggregate sometimes. You can have adhesion to various points, and then you can also have interception. So these are the main ways, together with size exclusion, that you would use. So it's a multimodal filter, okay? And the design criteria are a little different to multimodal depth filters used in the biotech industry, but it's a depth filter. A couple of other things to bear in mind with this is that this is a very mature industry. Today, leukodepletion is routinely done. These filters are quite cheap, actually. So it's an industry where cost is the main driver for change. If you can design a leukocyte filter that is, 80% the cost of current ones, you'll have a market. Okay? So cost in this case is important. It must meet specifications for red blood cell recovery and leukocyte clearance. This is typically 10 to the 3, okay, leukocytes you have to remove, and you're going to recover at least 95% of red blood cells. Otherwise, there's no point, okay? The process must fit into blood banking practice, as I said. I showed you how you're hanging these up and going on. If you come up with a process that requires somebody to watch this for 10 minutes, it'll never work, okay? And then you want easy to run, operating procedure, time taken for filtration. These are all important. Now, a couple of things. There's a lot of debate, and there are two ways to do this. You could do leukodepletion at room temperature. You could do leukodepletion at 4 degrees Celsius. Typically, red blood cell concentrates are stored at 4 degrees Celsius. And so there's a whole protocol for this. Once you donate your blood, which is at 37, 38 degrees Celsius, how long you keep it at that temperature before you cool it, okay? But once you cool it to 4 degrees Celsius, you don't increase the temperature again until you go to re-infuse. Too many temperature cyclings is not good, okay? And so the question is, how long do you have? If you're donating your blood in a blood bank, okay, they have all the equipment there, so it may be feasible to immediately take this leukodeplete at room temperature and then put it in the fridge at 4 degrees Celsius, okay? If you are doing a remote location, they'll typically collect it, put it at 4 degrees Celsius, ship it, and then do the leukodepletion. It doesn't matter, but there are two differences. You know that the resistance to flow through these membranes depends on a number of things, including viscosity, right? And the viscosity is going to be much, much higher at 4 degrees Celsius compared to 20 degrees Celsius. In addition, if you do it at room temperature, if you don't have a fairly regulated room temperature, okay, then you may get good performance, you may not, depending on what the filter is rated to run at, you see. So these are just some of the practical things that come into leukodepletion and how they are run. And so there are filters for 4 degrees Celsius, there are filters for 20 degrees Celsius, and so forth, okay? Okay, any questions on leukodepletion? I think that's, again, another very interesting takeoff on depth filters that are used in the biotech industry. Okay, let's move on then. We have a couple more things to talk about. So dialysis, I believe Professor Jacob covered, so I'm not going to talk about it at all here. He called it hemodialysis. So dialysis is a more general term. Dialysis is also used in the chemical industry, okay? So it's not just hemodialysis. Just want to point that out. So when you're making chemicals, say pharmaceutical manufacturing, from synthetic chemistry, not biotech, synthetic chemistry, you often have to take out components. You need to dialyze them out. Okay, so dialysis is used in the chemical industry. So that means that I want to talk then, before we finish, on something called hemofiltration, which is an ultrafiltration process. Okay, so did this move? It did. Okay, so as discussed, the driving force for hemodialysis. Well, I think it was discussed. I'm sure the driving force for hemodialysis is going to be a chemical potential difference, right, between the components. It's a chemical potential difference. It's not a pressure driving force. Okay, so at low rate, sorry, why did this R get cut off? At low rate, ultrafiltration, pressure-driven process used for water removal in hemodialysis, development of high permeability, large pore-sized membranes has led to new options for treatment. So, even in dialysis, you're trying to remove water sometimes, and that is done by putting a pressure difference, so you can increase the rate of transfer. If you're transferring by a chemical driving force, typically the flux is low compared to a pressure driving force, okay? Now, today we have high flux membranes. So the question comes up is this: in hemodialysis, the process might be too slow. It might be slow. You don't want the patient sitting there forever on the dialysis machine, okay? So for small species, urea and other things, small molecules you're trying to remove, that's fine. You can do them quickly. For mid-range, so we're talking about several hundred kilodalton, there may be contaminants you want to remove. Removing them by a dialysis process is slow because this is effectively a dense membrane, right? They have to diffuse through this. There's no convective flow through. But we know ultrafiltration would be fine, right? Apply pressure driving force, have the right cutoff for the membrane, you get it out quickly. So that is what hemofiltration is. So hemofiltration refers to a pressure-driven separation process where mid-molecular weight species, several thousand kilodalton, are rapidly removed by convective flow through the membrane pores. So it's an ultrafiltration process. Okay? So please remember, hemofiltration and hemodialysis are very different. They're doing the same thing, but they're very different because one is ultrafiltration, one is dialysis. One has got a pressure driving force, the other has a chemical potential driving force. Okay? Theory of ultrafiltration we discussed in earlier lectures, so I'm not going to talk about it here. So instead, here's a hemofiltration unit. And so this is just like an ultrafiltration module designed for hemofiltration. If you look, here's your blood, and here is the convective removal of components you don't want from the blood. Okay? And you can pump through it. Now, there is no dialysate, okay, as you can see. However, you do need to think of something. There's something here called replacement fluid. There's no dialysate. So in dialysis, you have a dialysate. You have to have a dialysate, right? Here, ultrafiltration, there's no dialysate. But in removing these species, you're also removing solvent. And solvent is water and other plasma proteins, okay? So you've got to put the water back for sure. Otherwise, the patient is going to lose blood volume. So you've got to put a replacement fluid. The downside with hemofiltration is you're also removing small molecular weight species that you may not want to remove, right? So you're going to have to find a way to put them back in or put replacements, synthetic replacements. You're going to have to do something, okay? Otherwise, the osmolality of the blood will change. You'll be losing essential amino acids, and there's a problem. So hemofiltration is good if your contaminant, what you want to remove is a mid-molecular weight species. But the problem is you need to worry about the replacement fluid and making sure that you don't cause more problems for the patient, okay? Okay. So these are some of the units that are used. And you can see it here. So again, here's the hemofiltration module. It's a bit like the one I showed you for cell washing. The patient is there, you hang it up here, a completely closed tube set, and you run it. So this is a membrane process. It's not a centrifugation process. It's ultrafiltration, okay? And then this is another schematic of what can be done. But this was a little different. See, this one is showing you a dialysate plus filtrate. And here it's showing you, basically, this is your blood that's going here. So what happens here? Well, that is the following. So, people have been working on hemofiltration and hemodialysis. And in many ways, these are like two different extremes. Let me go to the next figure. Yeah. And then I'll explain that figure. So in hemofiltration, maximum clearance determined by blood and filtrate flow rates, and with the substitution fluid is added before or after filtration. I showed you the substitution fluid at the end that time, right? Solutes with molecular weights of several kilodalton are removed at the same rate, whereas in hemodialysis, there is a monotonic decrease in the rate of removal with molecular weight of the species. The higher the molecular weight, the lower the rate of removal in hemodialysis. In hemodialysis. Hemofiltration, it should all be the same because it's convective flow. So hemodialysis provides superior removal of low molecular weight species such as urea. Hemofiltration and hemodialysis represent two extremes, right? One is removing at high rate mid-sized species. The other is removing at high rate low-size species. One is removing lots of solvent, which you have to replace. The other is not removing much solvent, but you still have to have a dialysate to put things back, okay? And so the development of high permeable membranes for dialysis led to the development of new treatment modalities where hemodialysis is combined with control rates of ultrafiltration. This is sometimes called hemodiafiltration, and that's that module that I showed you, where you have both a dialysate and you have convective flow. So if we go back to that figure that I showed you, yeah. So you may be able to run here some sort of combined process where basically you're using a membrane here. It has pores, but not as big pores as you would in hemofiltration, somewhere in the middle. And so there is some dialysis occurring, but there is also an ultrafiltration flux. So there's dialysate and filtrate. So you do have a pressure driving force across this, and you get so-called dialysis flux and ultrafiltration flux. And in the literature, this is discussed a lot, okay? And depending on the membrane, you can change that, okay? And so that's a kind of a compromise. So that way you don't lose quite as many of the smaller molecular weight species. You don't lose as much solvent, but on the other hand, you start to get slower rates of removal of the mid-molecular weight species. Okay? So I just wanted to point out that hemofiltration is an ultrafiltration process, and it is often complementary with dialysis. The one that's used really depends on the patient's conditions. Okay, what are you trying to remove? Okay, so in hemofiltration, greater amount of replacement fluid is required compared to hemodialysis. A combined process will minimize this limitation while providing high rates of removal of both low and mid-molecular weight species. Many names have people have come up with this for this process, one of which is hemodiafiltration, but there are others. There's like high flux dialysis and so forth, okay? Depends on the company and how they're marketing it. So I think we're almost finished. Hemodialysis, hemofiltration, hemodiafiltration are all mature technologies, all of them. Cost of healthcare is a major driver for cost reduction. So come up with something cheaper, people will use it. A couple of other things though. Dialysate is typically not produced from water for injection. Thus, there's a possibility of transfer of endotoxins to the patient. So what is this? So you know the dialysate fluid, you could use water for injection. This is water that is highly purified that you would use in a drip or something else. The industry doesn't use that, okay? That's just the way it's developed, and today there is no motivation to do it. It's just going to cost a whole lot more. So they don't do that. So the problem is you can have endotoxins. I wasn't sure if you knew what endotoxins are. So endotoxins are liposaccharides found in the cell walls of gram-negative bacteria, which can induce inflammation and fever as an immune response in humans and higher organisms. So there could be endotoxins in this water, okay? And they will not be there because it's not water for injection. And in the dialysis process, as the stuff you want goes into the dialysate, components of the dialysate go into the feed, right? It's a two-way process. You're matching the chemical potential. What goes in the feed is typically what you want to go in there, but if there are endotoxins present, they can go in there as well. So there is actually the one area of research in the area of dialysis membranes today that's of significance anyway, is trying to come up with adsorptive dialysis membranes that will selectively absorb any endotoxins in the dialysate. So as these things are going through, you'll be removing them, okay? But that is an area of research. How successful it is, we'll wait to see, but that's what's happening there. So I think that brings me to the end. Yes. So we will take our break now. I think it's just on 11 o'clock, yes? And I need to speak to Noel. Is he around or is he? Okay, because we're going to look at the homeworks and then we'll start after that.